Welcome everyone to BrainMap. As you all know, BrainMap is co-sponsored by the P41 Center for Mesoscale Imaging, housed in Martinos. It is my great pleasure today to introduce our speaker, Dr. Wang. Dr. Wang is an assistant professor of radiology at Harvard Medical School and Massachusetts General Hospital. Her research focuses on developing novel optical imaging techniques in combination with MRI and mathematical modeling to investigate the structural basis for the complex functions in normal and diseased brain, especially mapping the structural connectivity by preserving microscopic precision of neuroarchitectures. Dr. Wang completed her PhD in biomedical engineering at University of Minnesota and conducted postdoctoral work in neuroimaging at the Martinos Center. She obtained her bachelor's and master's degree in Huazhang University of Science and Technology, China. And we are very glad to have you here, Hui. Thank you so much. So let's thank our speaker first.
Okay, thank you very much for the invitation. It's my great pleasure to be here talking about our recent progress in microscopic connectivity by using an optical imaging technology called optical polarization sensitive optical coherence tomography. And the goal here is to use technique to image large-scale brain samples to really better understand the connectivity. Okay, so we all know at the center, we all know that understanding the neuroanatomical foundations for the circuitry is very important. It's important to study the normal functions in the brain disorders because it all lays the backbone of the neuron communication. And this effort has been recently emphasized by NIH Brain Initiative Connects Network that involves multiple grants in different species, aims to build brain-wide atlas. One particular emphasis is human and non-human primate circuit connectivity with the resolution of axonal fibers. So what are some challenges of the current technology to image connectivity, especially in the human brain? So the picture on the left is a very beautiful picture just published about 2 weeks ago, image a 1 millimeter cubic human brain cortex by electron microscopy. The nanometer resolution advantage really allows every single neurons to be mapped in this 1 millimeter cubic cortex and see where they go. Each axon, where they extend, where they go, where they terminate. But if we project that into a whole human brain, that is actually just this dot. It's less than one, yes, Nela. There are different layers of cortex. So yeah, so just say if you project that into the whole human brain, it's actually just this dot. It's less than 1 out of 1 million volume for the human brain. And this image itself took a year to accomplish. So if you just do your math and think about how to image the human brain, that's an impossible mission almost. On the other hand, the other side of the spectrum that we have the diffusion MR technique that image the whole human brain in vivo and both in vivo and ex vivo. They look at big fiber bundles of brain-wide connections. And it revolutionized our understanding of regional connectivity, but also it bears limitations in regions that multiple fibers converge together and then going into different directions, because within one voxel, you can see thousands of fiber bundles inside it. Okay, in the middle, there are optical imaging techniques that has microscopic level resolution and can image the fiber tracks directly. One of them is called polarized light imaging. That's a label-free image technique, can image the fiber orientations using a intrinsic optical property called birefringence. So here it shows a polarized light imaging of one brain, one slice, that the color shows the orientation of the fibers. And when you zoom in, you see that in this slice, this technique depicts the orientation of the fibers very nicely following this coloring. And the problem of this, as other optical imaging techniques, is that they image individual slices, thin slices of tens of micrometers. So when you look at the slice individually, they show very nice pictures of fibers. But when you like put the slices together, then you start to see enormous distortions between slices. That from this view, this is a stack of the slices put together from the study. And then across slices, it's very hard to track the individual fibers continuously because of this distortion. The fibers are shifted between, on the histological images between slices. So that imposes a challenge in 3D reconstruction of fiber pathways. So during the past years in our lab, we tried to develop the technique that can image the fibers with micrometer scale resolution and brain-wide coverage. And very importantly, we really target to reveal the connectivity in undistorted 3D space. And also, this technique should be compatible with histology-based neuroanatomical methods. Like in this pipeline, the sample can also be imaged with traditional histology. So that's our goal. And the technique we focus on is called optical coherence tomography, short for OCT. So OCT is an optical interferometry technique that gives depth-wise information of your tissue. And with a raster scan XY, it can reconstruct a microstructure in volumetric fashion of the human brain. And the advantage of that is it's label-free. It all uses intrinsic optical property, doesn't require any scan, and the resolution is close to histology, usually at 1 to 10 microns. And with one light shining from the top of the tissue, it can penetrate, see the information down to a few hundred microns in ex vivo human brains. So it's good to see that with the OCT, it can see the gross anatomy of the brain structures like this right brain, but it also has signals from everywhere because the signal is backscatter, everything scatters in the brain tissue. What about the targeted fibers we want to see? So we take another step down to use another optical property called birefringence. It's an intrinsic optical property again. It's caused by a regular arrangement of molecules in anisotropic structures. So think about the brain, a neuron that has axon and surrounded by myelinated fibers, by myelin sheaths. And the myelin forms a very regular arrangement along the axons, such that when the polarized light passes through these axons, and it actually sees two different refractive index along two axes perpendicular to each other. And after the polarized light passes through these fiber bundles, there results in a phase shift between the two orthogonal polarization channels. And this phase shift results from the birefringence that's defined by the difference between the refractive index along those two polarization channels. And this phase shift causes a phase retardance that's imaged by, we'll see, PI polarization sensitive OCT later. And it also results in an optical axis that's in parallel with this fiber orientation, the axon orientation. So by using that, we just add several polarization control like components into the traditional OCT system. And in the end, we detect two polarization states. And that, with that, we are seeing two additional optical contrast. One is called birefringence, a retardance that highlights the white matter in the brain. Here is a human cerebellum. You can see the regions of the white matter in the middle that's highlighted by the birefringence. You can even see like the fiber branches that go to individual folio. And using another contrast called optic axis orientation, resulted from birefringence as well. You see the fiber orientation in this plan. And that's explained by this color wheel. And when you zoom in a very small region, like here, around one folio, you start to see the different orientations from the optic axis. Okay, so with this technique, we can immediately apply a traditional tractography tool to study the connectivity. In the slides, for example, if we put seeds here, and then you can follow the fiber orientations and see different fiber tracks, different trajectories, some splitting from one point and some converging at points at like between the deep nuclei and to the deep white matters here. So, yeah.
Like, look at the yellow middle picture. It's like thin and then it gets thick. Is that just a 3D? You could think 3D a total conservation volume. Or is it that there's actually greater separation between the axons, right side of the picture and side? But what's accounting for that two things? What say track that has certain words or lots of times often that's why it's
yeah, that's a good question. I sort of think that's kind of a like limitation of the traditional tractography tools. So here it just shows like maybe in this region, that's a small seed that you start. So basically it shows like in this region, more fibers following this direction. And then the tractography will just follow that. That's my interpretation. I'm not 100% sure if it really preserves that. It spreads out more or like it's
because that I assume is a single 2D image.
Yeah, that's a single 2D image. Yeah, it's also possible that there are other fibers coming through the plane and turns and then yeah, it's possible.
See that.
Right. Yeah. So that's the limitation that about the traditional PSOCT, and that exactly leads to the discussion, the next part of the work, that's the 3D axis. But before going to that, we're so far we're showing like one slice and how to make it large volumetric reconstruction with the PSOCT system. That we add a vibratome tissue slicer into our imaging system, because PSOCT image from the blood place from the top of the brain and collect light back scattered to the system again. So we can image the top portion of the sample like this slice here. And then we can slice off what's being imaged and get the deeper regions exposed again. So this is the next portion. and then slice again and image deeper and slice off again until we finish this whole volume. And because two adjacent sections in Z has certain overlap between the two, and those can be used as a landmark to register slices, multiple slices. And because the image is done in this intact tissue format, so that there's no distortion. And in the reconstruction, we can just directly select the slices, imaged sections together to form a 3D. So we start, we image a human brainstem. This here it shows a block face of orientation image in 3 centimeter cubic brainstem samples, and you can see how the fiber orientation changes over different depths. And with a 3D reconstruction, we can clearly see the fibers continuously following this smooth trajectory over all the orthogonal XY, YZ, XZ planes. So here's kind of the evolution of the technique. This is 2014 when I first started here. That's the first system I built from scratch. It's probably made from hundreds of elements. And now we're just using a more compact system that we put the microscope, the stages, and the vibratom that's here together, and with our fully automated acquisition software, and this can go on and on for multiple weeks without stopping and human intervention. So, okay. So like Bruce said, that we're showing fibers, so far we're showing fibers in the plane. That's 2D, but in order to recover the connectivity fully, we actually need a 3D fiber orientation. So with traditional PSOCT, it has one illumination that's perpendicular to this tissue surface. And we're actually looking at a in-plane orientation, we call it theta. That's a 3D axis of this fiber projected to this XY plane. And this is what traditional PSOCT got in the optic axis, that's the theta. But what we really want to know is additional information that's alpha, we call it through plane orientation, because we need to know both in-plane and through-plane orientation in order to recover the 3D axis. So the way we did that is we got another measurement by tilted illumination. So we tilt the objective by a tilting angle, and then we get another optic axis that's a slightly tilted plane with respect to the original optical axis plane. And we get another measurement of a optic axis and along with birefringence. And we did a optimization to minimize the estimated birefringence vector that includes a birefringence value and the 3D axis orientation, and minimizing the errors between the estimated and the measured one with the two measurements. And we can recover this alpha, that's the inclination angle of the fibers. And along with that, in parallel, we also estimated this we call the true birefringence that removes the dependence of the birefringence to this alpha factor that reveals the real myelin content. So with the method, we did calibration of that, how accurate and how reliable the method is. First, we have a corpus callosum sample with manufactured it in a way that we can control the through-plane angles very nicely. We put a 30-degree edge, wedge across, and then that way we can make the through-plane angle plus minus 30 degrees with the original through plan orientation. But the implant orientation, we didn't change across the three setups. So that's our estimation through the algorithm. The implant, they remains the same. The through plan, you can see they show different colors. And when you look at the angle, the angular difference is 30 between the 0 and plus minus 30-degree setup. And importantly, we also found that we can accurately review the sign of the through plan orientation. In the next one, we want to know how big the range we can measure through this through plan axis orientation. Because you can imagine as we tilt the light more and more, we start to have less and less light back scattered to the camera, collected by the camera. So as SNR reduces, the accuracy might reduce as well. So we want to know what's the range this through plan axis orientation can cover. So we do the same trick of the corpus callosum again, put it with a different through plan angles artificially with a step increase of 10 degree in one experiment and 20 degree in another experiment. And we fit the preset through plan orientations with the estimated through plan angles. And we get slope very close to 1. That says that with this full range of through plan orientations, we can almost recover the real through plan orientation of the fibers within the 0 to 90 degree angle. And by combination of the implant through plan orientation, we can be confident that it almost covered the full spherical space of the 3D fiber orientation. So,
limits on the steepness of the angle that this technique will work?
Yes.
Like 50 upward.
Yeah, so the 50, because we tested, the estimation goes up to like 85, 86 here at the highest point. But because the original setup, like with 0 degree, there's already a kind of a small through plan angle, not 0. So it adds up by tilting 50 degree, it adds up to like 85. I bet at 90 degree that there's no signal at all. So we don't know that, but almost within this sphere can measure the orientation. Okay, so then with this method, we go on to test in a whole coronal section of the human brain, and this is the implant orientation, and the next figure is the through plan orientation. They all use the same color wheel, and it's interesting to note that deep in the white matter regions, deep down the south side, and you see multiple fiber bundles meet together. And usually in the space, you can see that there's a fiber bundle directly coming through the plan as well. So here you can kind of think of their fiber intersections from all different 3D space here. And this one is part of the corpus callosum that really stands out, showing very fine structures of the fiber bundles. And we combine the two orientation measurements into 3D axis representation and compared with a diffusion MR scan that's done by anesthesia here. And we found they are roughly good agreement for at least the big fiber bundles here. In the next thing, we really want to take advantage of the resolution by PSOCT here to image a more complex structure. That's the human brainstem. And this one shows a section of the medulla region. That's the first grayscale image is the birefringence, basically tells you how much myelination there is. And then the next two are the in-plane and through-plane orientation. And the last one is the 3D fiber orientation in the brainstem that you kind of see they group into several different groups, like part of the fibers going left and right, and part of the fiber going through the plane, that's the blue one. That's axial direction connecting the brainstem to the spinal cord to the majority part of the cerebral. And with Carlin's help, we can see that we can really segment the small nuclei and the fiber bundles in the brainstem, and that can be very useful for tracking later. And just as a comparison, this is a diffusion MRI at 300 micron resolution, which is already very nice compared to the in vivo technology. But yeah, so you can see the difference. Okay, so by using the 3D axis orientation obtained by PSOCT, we can actually conduct a tractography analysis on those reconstructed volumes. So here, Carlin again helped us manually segment all the fiber tracks, fiber bundles, and nuclei, and we construct the tractography. Here, I'm just showing zoom in version of 3 bundles. One is closed surrounding this inferior nuclei, and you can see that the fibers are going into different directions, trying to intervene with the nuclei in the middle, nucleus in the middle, and we put a small ROI here. You see the fibers spread out and put a surrounding like this fiber structure. And the other thing is this is the pyramid that goes really mostly through the axial plane, up and down. And you see on the XY, you're seeing the axial plane, they are kind of crossing, interweaving with a small angle. And in the XZ, you look at the longitudinal direction, you kind of see like the fibers are twisting or interweaving with a small angle. And here's another bundle that shows similar interweaving pattern, but with a different angle. They're crossing at a more larger angle, sort of like this pattern. So those are some of the fine structures that we revealed by this connectivity study that we'll do more validation on.
So I'm saying that 2 and 3 are, in regular tractography, you would be used to seeing a consistent color, right, for those tracks that belong to the bundle. And here you see, like, the colors still indicate the color?
Yeah, they do. Yeah, so it's like the traditional diffusion tractography color, like RGB.
There's a lot of colors here, so are you saying because of this nothing behavior, that's why you see all sorts of colors?
You mean like this one? That seems to be, yeah.
Right?
Yeah. I think they're just
like there you see very
like yeah, I think they are really represent the different fiber bundles. They all turn to go into, for example, in 2, they all turn to go into superior inferior, this direction in general, but they don't like go parallel in this direction. Instead, they kind of doing this thing to go up and down. That's the detail that's revealed by the PSOCT.
How are you going?
That's a very good question. So far we don't have validation yet because the only knowledge we know is from like histology, the neuroanatomy, like how are the bundles going from like histology study. But really regarding individual fiber bundles, we don't really know. But if they really go this way. Yeah, that's the thing. But we are getting there for a true validation, like in the new project. Yeah, yeah.
Slabs that you're shaping off.
Yeah, so that's exactly leads to the the one that I'm going to talk about.
It's too interesting. Just, yeah, there's a lot going on there. Exactly. This kind of braiding pattern I'm talking about, that is there any precedent for that elsewhere, or is this like a novel discovery?
I mean, I'm sure like neuroanatomists know that fibers are kind of twisting and like interweaving with from different areas into a very compact bundle. So for example, pyramid, we're talking about very compact fiber bundles, right? There are neuroanatomists know better than me here. But I believe that is very compact fiber bundle, and they kind of form like small groups that travels kind of navigating, not directly, but through this bundle. So I think it's known in general. No, this is just a, because I can't show this with the current tractography tool. So this is my interpretation. Yeah. Wait, sorry, what's that?
What's left of the
There's two cartoons. Yeah, those are the two cartoons, like my interpretation for this fiber configuration.
If it looked like that, it would show up in your table.
Yeah, this is the vectors that of the vector orientation in the data. Yeah, but it's just the tradition, the current tractography, it's all like if we do a tractography along that, like what's shown in two, they look correct at this zoom out image, but when you go in, looks like the fiber is doing this, like a snake snaking this way. But what really happened, I believe, because we also see the cross sections of the fiber bundles in the intensity image and orientation, that they really form like this.
If it has anything like this kind of gradient or spiral, you would expect, you would have a periodicity rather than some kind of random action. Is that visible in the data?
So yeah, I think it's here, right?
People on the Zoom sometimes are not hearing.
Okay.
No, it's okay. If you could either repeat, or people could just, if you could just speak a bit loud, shout basically, that would be great. Sorry. Yeah. So Bruce's question is really how to interpret this fiber. Do they really like show in this pattern, right? Yeah. So I think it kind of shows this periodical features, right? If you look at orientation closely, well, you don't need to look at it's probably too small for individual vectors, but the color really shows a pattern of periodic kind of color change. So that made me sort of think it may be like this configuration. That's my speculation based on this data. But I think the histology image before showing like the small fiber bundles in the big small fiber tracks grouped in the big fiber bundle, if I understand that correctly.
Yeah. So why is one so different?
One is just because that's the inferior alveolar nucleus. Yeah, that's a nucleus, and it's like deep nuclei in cerebellum that has a convoluted sheet of gray matter, and then they're surrounded by the white matter. So that's why it's here. It's
but
here.
It just doesn't seem that any of those tracks have any of this.
Yeah, because this is this is immediately like this structure right here. Whereas the other two are from here, they really like from the spinal cord and goes to really different brain regions in the cerebrum. So I kind of assume they go together and travel to their like destination. I don't know, all this warrants further validation and investigation, but this is the observation. Yeah, so this is 10 by 10 by 100 micron. Yeah. Okay, yeah. Yeah. Yeah, yeah. The ultimate determination is called the optical diffraction. But usually it depends on, for OCT, it has axial resolution and lateral resolution. They're decoupled. The axial resolution depends on the bandwidth of your light source. The broader bandwidth of your light source, the higher axial resolution you can see in depth. And the XYZ lateral resolution is determined by the objective lens you're using. The higher magnification, the higher lateral resolution. Yeah, so here is also, but there's also trade-off, like you use higher magnification lens, you can see higher resolution, but you also lose your depth of focus, which means within a depth range, all the image is in focus within that Z range. So the higher magnification, your focus is sharper, so that you end up seeing less focus in Z. And then you go to like do a Z stack, like traditional microscope, and that's really increase the imaging times. We can do higher resolution than this, but this is kind of a trade-off between the volume and the resolution.
One comment actually from the audience. So it says, there's a very high resolution DMRI in marmosets published, which shows this same pattern in the corticospinal tract.
Okay, cool. We'll have to check it out. All right, thanks. Okay, yes.
I have a related question since somebody brought up the marmosets. I had been thinking about this because at the beginning, you mentioned the potential of like using a method like this to do cross-species multimodal studies. And I was wondering, so we're looking at human brain data, I was wondering comparatively if you have looked at macaque and mouse data. And because with mice, I imagine that even though at this really, really tiny resolution, it might take you forever to get just this data in humans. With mice, it must be feasible to just do a full reconstruction in this way, right?
Yeah.
So how do they compare in terms of the total resolution and the amount of time it takes you to take the data, and then the similarity of the fiber tracks across the species? I'm sorry, that was like 3 questions in one. You don't have to answer all of them.
Yeah, one big question. Let me talk through the next study. Some of the questions will be answered here.
Okay. Sorry.
Okay, so yeah, so just the PSOCT itself shows some interesting findings that led us to do a more comprehensive study that really use multiple imaging methods to look at the connectivity together. And this study are funded by 2 NIH Connect grants. One big one is the LINK, the UM1 that anesthesia lead. And another one is Brainstem Connects that Bruce and me coded together. So in this part, that like Bruce asked, how true the fiber connectivity really is, right? Revealed by PSOCT. So in the past 5 years, we established a multimodality pipeline that we image from a whole brain by ex vivo MR or diffusion MR, and then we do PSOCT and slicing. And the slices go to a traditional histology, and that can be cleared, scanned, and then do a cross validation. Yeah, that part is very nice pipeline we just published last year, but there's still like this, once the slides are cut off and mounted on the glass slides, there are enormous distortions And you have to do a nonlinear registration between the PSOCT image and the histology images, right? And we really, and that's very, that we have very good preliminary results, but still, if we want to do a voxel-wise comparison for the fiber tracks in say 3 microns, that's still pretty challenging. So what can we do better? One way we came up in this link connects grants is that we can use ground truths of neuronal tracers from monkey brain. So traditionally, our collaborators, Suzanne Haber and anesthesia, they inject neuronal tracers into monkey brains. And then they do diffusion MRI and they do like slices of those tracers and image where the tracer goes from slice to slice so that they can establish these connections of fiber tracks across multiple sections. But they are like there, they only look at where this tracers goes across those slides and find the corresponding locations and establish these different tracks. And here you kind of see that the tracks going from different cortical regions and kind of all navigate together into this very small space in the internal capsule. And that goes further diverge later downstream. With a large, yeah. Left is tracer, so they inject tracer from different cortical regions like here, and then so the tracers are fluorescent. They can look at where the tracer goes. That's light up in each individual like slices. It's slice-based. They find corresponding locations. And then from there, they can like make this trace schematic here showing like which fiber track, what's the trajectory of the fiber tracks by following the tracer locations. Okay, so with light sheet microscopy, we can actually see individual axons with much higher resolution. And this specific setup called SCAPE, light sheet microscopy, actually use a single objective with a tilted imaging plane and collects fluorescence in this tilted plane. And if we scan this tilted plane laterally, it actually collects this volumetric imaging. And since the tracer is fluorescent, we can actually use the light sheet microscope to collect the tracer imaging with individual axonal resolution to say that in 3D. And very nicely, because this configuration is actually very similar to PSOCT in a way that it images the block face. It shines light from the top and collects light that fluorescent back to the objective and collected by the camera. It can be integrated into a imaging and section, this scheme, right? So that potentially it can create a large volume of like fluorescent images with the neuronal tracers. So here's one like example showing this is a slice, a macaque slice that has tracer injected in, and Elizabeth's group from Columbia, they look at the axons by their light sheet microscope like this, and they see the axonal bundles that's light up by the tracer. So like I said, like because both the light sheet and PSOCT image the samples block face from the top and collect light going back to their individual cameras. Oops, sorry, I probably need a charger. So Okay, so that you can imagine the two can be combined together, and so that all this imaging take in block phase, and they can be imaged at the same time, so that they have voxel to voxel. Thank you very much. Yeah, this works. Good. They have voxel to voxel correspondence in the end, right? There's no registration needed because they image the same plane and then section it off. I image the same plan again by the two modality and section it off, and each one reconstructed one is undistorted in 3D, and they have direct correspondence. So the idea is very good, except the two technology are inherently incompatible. The reason is the PSOCT, it's really image the myelin in the brain, that it also needs a scattering. So we need to preserve the myelin to have both scattering and birefringence property. Whereas light sheet microscope, they usually image transparent tissue, which means the tissue is cleared. What's being cleared out is mostly myelin, because the lipids in the myelin generates most of the scattering signal that gives troubles to the light sheet microscope. So the two system hardware-wise can be put together, that makes sense, but this tissue processing procedure is really a headache for us. So our collaborator Zhu Hao from Cornell Medical School, he's a chemist. He's very good at like refractive index matching and clearing, came up with a solution. What about we do a partial delipidation to the brain that only removes the lipid from cell membranes, but preserve the lipids from the in the myelin, so that we still have the myelin, the birefringence that's needed by the PSOCT, but we also do a partial clearing that increase the light penetration, good for light sheet microscope. So we tried different procedures, and we ended up finding one that really preserves a good PSOCT information. This is a mouse brain, and the same mouse going in the same procedure, we look at light sheet on the light sheet microscope and see the fiber orientation, the fiber information there. There's no orientation, but the fibers really lights up at a much higher resolution. This is a zoomed out one, but they do show individual axons if you zoom in. So then with this problem solved, we're making progress in combining the light sheet microscope with PSOCT. Yeah. Yeah. Yeah, yes. Right. That's kind of the sweet spots that PSOCT preserves the signal, whereas light sheet microscope can also get the signal from relatively deep regions. So yeah.
But you don't have any research showing how else.
Yeah, I don't have it here, but we do show like we use different level of delipidation. Complete delipidation, there's no PSOCT orientation signal there at all. Even this one, from the intensity, you kind of see they don't have much like differentiation in the brain. So it's been like the scattering property has been matched very well within the brain, but the birefringence is largely preserved for this process. Okay, so we're in the progress of setting up the combined system. Stay tuned and come back in a year, see the results. So then I probably just want to skip this one just to see over the past 10 years with anesthesia, we really try to use the PSOCT to validate and guide diffusion tractography method, because like you all know, the single voxel is very big, that includes multiple fibers with different orientations, then you have no distinction between crossing and casing from different directions. So we showed that with PSOCT, it helps to characterize different acquisition method and reconstruction method to know their errors better, as well as the resolution requirement for the diffusion MR to see the tracks correctly. And the very last few minutes, I'm just going to briefly show the PSOCT usage in brain development. So for people who are doing pediatric imaging, we all know that infant brains are not just small brains in size. The brains are highly underdeveloped, and the current imaging techniques have challenges in imaging this pediatric brains. So similarly, this unmyelinated brains in early life that also impose challenges for PSOCT because of the low myelination cause low birefringence, and PSOCT relies on birefringence to see the fibers and the fiber orientations. But on the other hand, it also creates an opportunity for us to study the development of myelin in the brain, because we show before that the optical property has been proved to be correlated with myelin content. Those studies are in adult brains, studied different brain regions with different levels of myelination. But for this study, we used a PSOCT and diffusion MR techniques to investigate the white matter development in brain samples across the first 5 years. So this is a study in collaboration with Dr. Ami Takehashi. And from her lab, we got the brains from NIH Biobank brains that are imaged by diffusion MR already. And we collect one slab imaged by PSOCT, and we get the parameter maps and compare with diffusion MR. So here it shows 5 slabs, coronal slabs of brain samples from subjects with different ages. The youngest is 3 months old, 6, 15, and 2, 4 years, and 4.5 years old in the end. And this is scattering property, scattering coefficient, and this is retardance that represents the birefringence. So you kind of see like nicely across this timeline, the scattering property, scattering coefficient increase over the 5 years, and they have a big jump after 1 month. No, sorry, one year to the 4 years. And we also show the fiber orientation image of those 5 samples. We were interested to know whether this low myelination can still preserve the fiber orientation information in the young, very young brains. And we're very glad to see that the fiber orientation can be shown in like brain samples as early as 3 months old. And the SNR is just better and better as the brain develops. So those are some detailed comparison using still 2D tractography by PSOCT orientation to see different fiber configurations. And so with that, I will just summarize the talk, saying that PSOCT provides label-free measurement of fiber orientation at micrometer level resolution. And by incorporating serial sectioning into the block phase imaging, volumetric reconstruction can be realized without interslice distortion as seen in traditional histology. And 3D axis orientation is developed in PSOCT support the real macroscopic tractography. Combination of PSOCT and Latchit microscope enables precise validation of neuronal tracers in monkey brains. And lastly, the PSOCT imaging of mildly myelinated brains opens new opportunities to study neurodevelopment. So this is a whole picture of the lab. Connectivity is one portion of the lab projects. We have hardware development, multimodality, neural architecture studies, and vasculature in the lab. And I'd like to thank all my lab members, collaborators, and funding sources. With that, I'm open to questions. Thank you.